Clarity Clinical Solutions.
Imagine a clinical trial that is not a single experiment, but a perpetual testing engine, a trial that can add new treatments as they become available, drop ineffective ones early, and answer dozens of research questions simultaneously using shared infrastructure. This is the vision of master protocols, and it represents one of the most important innovations in clinical trial design in decades. There are three main types. Basket trials test one drug across multiple diseases that share a common genetic marker. Umbrella trials test multiple drugs against a single disease. And platform trials are open-ended master protocols where treatments can enter and leave over time. Together, these designs are transforming how we evaluate new therapies. Let us clarify the three designs. A master protocol is the overarching structure, a single protocol that governs multiple sub-studies, each potentially testing different treatments or patient populations. A basket trial enrolls patients with different disease types, say, breast cancer, lung cancer, and colorectal cancer, that all share a specific genetic mutation, and gives them all the same targeted drug. The question is, does this drug work in any cancer with this mutation, regardless of where the tumor originated? An umbrella trial flips this around. It enrolls patients with the same disease type, like non-small cell lung cancer, but assigns them to different experimental drugs based on the specific mutations found in their tumor. Each design serves a different purpose, and they can even be combined within a single master protocol. The difference between the traditional approach and a master protocol is dramatic. Traditionally, each drug-disease combination requires its own separate trial, its own protocol, its own ethics committee review, its own site contracts, its own data management system. Setting up each trial takes months to years. With a master protocol, all of this infrastructure is shared. One protocol, one institutional review board, one data system, one network of clinical sites. New treatment arms can be added in weeks instead of years. A shared control group serves multiple experimental arms, reducing the total number of patients who receive placebo. Ineffective treatments can be dropped early, and promising ones can be expanded, all within a single, continuously running trial framework. The adoption of master protocol designs has grown rapidly. Over 100 master protocol trials have been launched globally, primarily in oncology. The Recovery Trial, the most famous platform trial ever conducted, enrolled over 40,000 patients across 180 UK hospitals and tested more than 10 different treatments for COVID-19 in just 18 months. Platform trials can produce results 70% faster than traditional trial designs for similar research questions. The number of new master protocols launched each year has more than doubled since 2020, driven by the success of COVID-era platform trials and growing regulatory acceptance. A basket trial is designed around the idea that genetic mutations in cancer can be more important than where the cancer originated. For example, if a drug targets the BRAF V600E mutation, it might work in melanoma, colorectal cancer, lung cancer, and thyroid cancer, all of which can carry that same mutation. Rather than running 4 separate trials, a basket trial enrolls all eligible patients under one protocol and analyzes response rates in each disease-specific basket. The most ambitious example is NCI-MATCH, which screened over 6,000 patients and tested more than 40 targeted therapy arms across hundreds of tumor types. Importantly, basket trials led to the first tumor-agnostic drug approvals by the FDA, drugs approved for any solid tumor with a specific biomarker, regardless of where the cancer started. Umbrella trials take the opposite approach. Every patient has the same type of cancer, but they are assigned to different treatment arms based on their tumor's specific molecular profile. I-SPY2 is the most famous example, a neoadjuvant breast cancer platform that has tested over 20 experimental drugs since 2010. Patients are screened for a panel of biomarkers and assigned to the most appropriate experimental arm. I-SPY2 uses adaptive randomization, meaning that as data accumulates, more patients are assigned to the arms showing better results and fewer to the arms that look less promising. This is more ethical than traditional fixed randomization, and it answers the question faster. Platform trials take the master protocol concept to its logical conclusion. They are designed to be open-ended. Treatments can enter the trial when they become available and leave when results are conclusive. All arms share a single control group. The recovery trial is the definitive example. Launched in April 2020 during the first wave of COVID-19, it tested dexamethasone, hydroxychloroquine, remdesivir, convalescent plasma, tocilizumab, and many other treatments. The dexamethasone result alone, showing a 35% reduction in mortality for ventilated patients, is estimated to have saved 1 million lives worldwide. Recovery cost about $30 million total, a fraction of the cost of traditional pharma-sponsored trials. Master protocols require sophisticated statistical methods. Adaptive randomization changes the probability of assigning patients to each arm based on accumulating results. More patients go to better-performing arms. This reduces the number of patients exposed to ineffective treatments while speeding up identification of effective ones. Bayesian hierarchical models borrow information across treatment arms, improving precision for smaller subgroups. And because multiple treatments are tested simultaneously, careful multiplicity control is essential to avoid false positive results. Statistical methods like gatekeeping procedures and Bayesian multiplicity adjustments keep the overall error rate at acceptable levels while maximizing the trial's efficiency. The FDA has been increasingly supportive of master protocols. It issued draft guidance in 2018 and final guidance in 2022 covering trial design, statistical considerations, and regulatory expectations. The guidance emphasizes that each treatment arm within a master protocol must meet registrational standards. The master protocol does not relax the evidence requirements for drug approval. Several drugs tested in master protocols have received accelerated approval based on single-arm substudies within the platform. However, regulatory complexity remains a challenge, especially when multiple drug sponsors are involved, each with their own regulatory submission timelines and data requirements. Master protocols offer significant operational advantages. A centralized institutional review board reviews the entire protocol once, rather than each substudy separately. Sites sign one contract covering all substudies. A single electronic data capture system manages data across all arms. But the operational challenges are equally significant. Governance: who decides when to add or drop a treatment arm? Sponsorship: which company holds the investigational new drug application for the master protocol? Data rights: how do competing pharmaceutical companies share control group data? Drug supply: coordinating shipments from multiple manufacturers is complex. And sustainable funding for long-running platform trials remains a persistent challenge. Recovery is the most powerful demonstration of what platform trials can achieve. It was built in just 9 days using the UK's existing National Health Service clinical trials infrastructure. It enrolled patients from over 180 hospitals across the UK. The trial tested over 10 treatments and delivered practice-changing results for several of them. Dexamethasone reduced mortality by 35% in ventilated patients and 20% in patients receiving oxygen, a result that became standard of care globally within weeks. The hydroxychloroquine arm enrolled 11,000 patients before being stopped for futility, definitively answering whether that widely promoted drug was effective. The entire trial cost about $30 million, a fraction of what a single pharmaceutical company would spend on a typical Phase 3 trial. Despite their advantages, master protocols are not for every research question. The statistical complexity is substantial, multiple comparisons, adaptive designs, and information sharing require sophisticated methods and experienced biostatisticians. The operational overhead of coordinating multiple sponsors, drug manufacturers, and regulatory submissions can be overwhelming. And master protocols work best only when certain conditions are met, when there is a strong biological rationale for biomarker-driven treatment, when multiple experimental treatments are ready for testing, when a shared control arm is appropriate, and when the research community is willing to collaborate. For simple questions, does this drug work for this condition compared to placebo? a traditional randomized trial is often the better choice. Looking ahead, master protocols are expanding far beyond oncology. Platform trials for Alzheimer's disease, autoimmune conditions, and cardiovascular prevention are being developed. The infrastructure created for recovery is being maintained as a permanent pandemic response platform. In the longer term, master protocols could create learning healthcare systems where clinical research is continuously integrated with patient care. Patients could be screened once at diagnosis, matched to the best available experimental therapy based on their molecular profile, and followed seamlessly across treatment arms as their disease evolves. This patient-centric vision, rather than drug-centric, represents the ultimate promise of master protocol designs. So here is the bottom line. Master protocols do not replace traditional randomized controlled trials. But they add a powerful new option for situations where multiple questions need answering efficiently, when there are multiple promising treatments, identifiable patient subgroups, and a research community ready to collaborate. By sharing infrastructure, controlling costs, and adapting to emerging data, these designs accelerate the pace of clinical discovery. They have already saved millions of lives through trials like recovery. And as they continue to evolve and expand beyond oncology, they will become an increasingly important tool in the clinical researcher's toolbox.